Taking an organ from one human being to give life to another is one of the greatest advances of modern medicine. It takes huge surgical skill and clinical know-how, but above all, it requires brave and difficult decisions by everyone involved. First of all, the donor's family must consent, still reeling from the shock of their bereavement. A year ago, Ashley's mum, Mandy, died suddenly from a brain hemorrhage at the age of 46. Mandy was signed up to the organ donation register, but in England it is the family that has the final say. Once an organ becomes available, it's a doctor's difficult decision who should receive it. With organs in such short supply, clinicians must judge when a patient is so ill they must go on the transplant list, and when they've become too sick and wouldn't survive the operation.
It really does matter. We can make a difference. And those that are on the list deserve a chance. And we try to get as many people on that list as we can. We can't save everyone, but we try.
There are thousands of people who owe their lives to pieces of dead pig tissue, and no, I'm not talking about the admittedly life-affirming taste of bacon. For the last 50 years, we've been replacing damaged human heart valves with pig valves. So if we can transplant heart valves from pigs, why not entire hearts, or kidneys, or livers, or lungs? How come you can walk around with a pig's heart valve inside of you, but you reject a kidney from another human? And out of all the creatures in the world, why pigs? So many questions. Transplantation of organs from one species to another is known as xenotransplantation, and it turns out to be pretty bizarre. But since there's a shortage of human organs, figuring out how to get a steady supply of animal organs and tissues that we could use is something we might want to figure out. Transplantation from human to human is tricky enough, much more difficult than matching blood types. The new organ is often rejected by the recipient because of a protein called human leukocyte antigen, or HLA. Almost every type of human cell has different kinds of these proteins on its surface, and normally they're the immune system's undercover agents, marking targets for it to attack. So if you transplant an organ from one person to another and the recipient's immune system recognizes the donor's HLA as foreign, it'll attack the transplanted tissue. This can lead to severe blood clots, failure of the new organ, and all kinds of painful symptoms that put together amount to transplant rejection. The problem with these HLAs is that they are incredibly diverse, with thousands of variants, which makes it very hard to find a donor whose chemistry matches a recipient. Some people spend years on the waiting list, others never get a match at all. But there are a few ways around the problem, and one of them is to skip humans and move to pigs. You might not find this too flattering, but you have a lot in common with a pig. Your immune system functions in a similar way, and your organs mostly do the same things. And pigs are the right size. You wouldn't want a heart valve transplant from a mouse or a beluga whale. The main thing is that a heart valve doesn't need to be alive to do its job. It's really just a piece of tough, flexible connective tissue that acts as a glorified piece of plumbing to direct blood flow. So if you blast the pig valve with a chemical called glutaraldehyde, you could preserve it while killing all of its cells in the process. Said cells can't produce those HLA proteins, so there's much less risk of rejection. The risk is not zero, though. These heart valves can wear out over time since they're dead and unable to replace damaged cells. Also, sometimes the patient's immune system still gets nervous about having this foreign thing in the body. The dead cells still have some antigens on their surface, and patients, especially those with stronger immune systems, can react to them. The valve may eventually fail. But some more extensive genetic engineering could help. For example, you could remove the gene that tells the cell to produce some of its other antigens, or throw in some human genes to make the tissue seem more familiar to the human immune system. They've tested these genetically engineered pig organs in non-human primates like baboons, which only survived for a short time, but the technology is advancing quickly. The downsides? Some people aren't comfortable with the idea of genetically engineering pigs to express human genes, even if it's only to trick the human immune system. Plus, cloning enough pig cells for the technique to work is mega expensive. For now, these genetically engineered pig organs would be too costly to reach any patients who aren't like at the private jet level of wealth. According to the National Institutes of Health, there are about 3,000 people in the United States waiting for a heart transplant. But every year, only 2,000 hearts become available. And human hearts actually have a lot in common. They each have 4 chambers, 4 valves, and they're about the same size. Anatomically, they are highly compatible with our bodies. Genetically, they are not. The biggest barrier to all organ transplantation is rejection, which is when the host's immune system attacks and kills the transplanted organ. Even human-to-human kidney transplants, the safest and most common kind of transplant in the world, have about a 15% rejection rate after 1 year.